Today we're going to talk about bringing a biotech product to market, and more specifically, the process for bringing a drug to the marketplace. This does not directly apply to your soda project, but the process will be similar for any genetically modified organism. And we are going to look at specifics for a GM food product later, but we're going to start with medication. So there are 4 main phases in this process. Today we're going to focus on phases 1 and 2 of the drug development process. So the number of biotechnology products marketed is expected to increase as the industry expands. Drug development is a significant activity in the biotechnology industry. Current products that biotechnology companies are bringing to market include drugs to treat rare diseases, vaccines, products that use genomic data to customize medical intervention. This is also known as personalized medicine. Genetically engineered mosquitoes that can reduce the spread of mosquito-borne diseases, or genetically engineered salmon that grow faster, and DNA forensic tests. Biotechnology companies work to bring products to the market that can meet consumer needs and about which there is sufficient biological knowledge. And the regulatory process is different for each type of product. And today, again, we're focusing on medicine. So today we're going to discuss the first 2 phases of the drug development process, and those phases are the discovery phase and preclinical research. Next time we'll talk about phases 3 and 4. This graph is really great because it shows roughly how long each phase takes. As you can see, the first 2 phases take about 6.5 years. So that's getting through drug discovery and preclinical trials. The second stages, or the second round of stages, phases 3 and 4, take quite a bit longer. But this is where we're actually testing on humans, where it says clinical trials. That takes roughly about 7 years. And then there's an additional FDA review process after human clinical trials before the drug can actually make it to the marketplace. Once the drug makes it to the marketplace, the FDA is still monitoring use. And so sometimes you'll hear that a drug was pulled off the shelf or it was pulled out of the pharmacy. And that's because they're monitoring it even after the drug is actually being sold. The process to bring a drug to the market is complex, risky, lengthy, and expensive. For every 5,000 to 10,000 new drugs, only 1 gets approved by the FDA. The time to take a drug from discovery to market ranges from 10 to 15 years. And the estimated costs of developing a successful drug are $800 million to $5 billion. The one reason for high costs is thousands of failures. Common problems include unexpected safety issues or a failure to demonstrate the drug's effectiveness. In addition, drugs must be manufactured according to the Good Manufacturing Practice standards, which are necessarily stringent, which means strict, and problems here also lead to failures. This presentation will present information on phases 1 and 2 of the drug development process, which are drug discovery and preclinical research. The first phase of the biotechnology-based drug development process is drug discovery. In this phase, researchers examine the biology of a disease in order to find the underlying cause. This knowledge is then used to consider how to change the biological component in order to treat the disease. For example, if a disease is caused by overproduction of a protein, then researchers would search for drugs that decrease the expression of the protein or increase its breakdown. There are 4 possible sources of drugs. Those include nature, chemical synthesis, recombinant DNA of proteins, and high throughput screening of hundreds of thousands of substances. High throughput means quick screening through an automated process for potential biologic activities. Testing each substance separately would take too long and be too expensive. Watch the following video.
My name is Elaine Donahue and I work in the high throughput screening group here at Novartis. High throughput screening is just a quick way to basically screen a biological reaction against a large number of compounds. In the very early days of drug discovery, in order to find a compound or look for a drug. It was really sheer luck. But now with the existence of high throughput screening, we're able to identify molecules that are active on particular targets or pathways of interest much more quickly to give us starting points for drug discovery. We couldn't do our job without the automation. The automation allows us to consistently and rapidly run these assays against our compound library. Our compound library is probably well over 2 million compounds at this point, and in the high throughput screening group, we screen about 1.6 million of those compounds through each of the targets of interest that come into our group. It used to be to run each test that you had to hand pipette into each well to run each reaction. Now with the new instrumentation, we have things that will, in the same time that you're pipetting into one well, can dispense into an entire plate and enable us to go from running 30, 40 samples a day, and now we're running up to hundreds of thousands of samples per day. What used to take months and years, we can now do in days and weeks. The robots allow us to run the assays around the clock. They allow us to run the assays in a very consistent manner. Sample 1 on the first plate is treated the exact same as the last sample on the last plate. To do this kind of process manually, you would never have such consistent data results. I think the most significant challenge with high throughput screening is the vast amount of data that we're generating now. When we used to generate 100 data points per day or even less, we're now generating 150,000 data points per day and sometimes even more. But then what to do with that data, that's where the scientists play a role. I think the main benefits for using high throughput screening drug discovery is the speed at which you can get to a drug. I think the motivation for all of this is to find something that can help a patient. And back in the day, it was a very slow process to get there. And now that we've enabled high throughput screening, we get a lot closer to starting points for drugs. We get there a lot faster than we used to.
If our drug meets all the requirements for phase 1, we move to phase 2, which is preclinical research. The U.S. Food and Drug Administration, or FDA, requires that candidate drugs be thoroughly tested before being used in clinical trials with people. During this phase, the biotechnology company carries out tests to determine the drug's physical characteristics, safety, usage, dosage, how to administer the drug to a patient, and its pharmacology, which is how the drug acts in the body. And the FDA oversees this entire process. For most drugs, animals must be used. Only animals can provide information about pharmacology and general safety. Pharmacology measures include the drug's persistence in the body, which in turn is related to how it is absorbed, distributed, metabolized, which means broken down, and excreted. However, the FDA has research and development efforts underway to reduce the need for animal testing and to work toward replacement of animal testing because of widespread concerns about the ethics of using animals. The next step in the preclinical trial phase is scaling up drug production. This is important preparation for the clinical trials. Drugs must be produced or manufactured according to the FDA's current good manufacturing practice standards, and these are updated regularly. You can look at the FDA's website and look them up. You can actually get a PDF of them. If the drug is a recombinant protein produced in a living cell, scaling up production presents the following challenges. Multiple iterations or repetitions are needed to determine methods and quality controls. It may not be clear which type of cell, it could be bacterial, yeast, or mammalian, will result in optimal expression and purification of the active protein. The exchange of gases and the distribution of nutrients becomes more difficult as the container becomes larger. Existing staff may not have experience with production methods, and contamination can occur. Before any clinical trial can begin, the researchers must file an investigational new drug application with the FDA, also known as an IND. The application includes the results of preclinical research, a description of the drug's composition and how it works in the body, how the drug will be manufactured, and a detailed plan for the third phase of drug development, which is human clinical trials. The FDA reviews the application to make sure people participating in the clinical trials will not be exposed to unreasonable risks. The clinical trial must also be reviewed and approved by the biotechnology company's Institutional Review Board, or IRB. The IRB is charged with reviewing all plans for human subject research in order to protect people's rights and welfare. Any institution that receives research funding from the federal government must have an IRB. So this was the summary of what has to occur in phases 1 and 2 of our clinical trials of our drug development process. Next time, we'll talk about phases 3 and 4, which include human testing.